Okay, good afternoon to everyone. So, welcome to today's class. We have started genetics, and yesterday we have discussed these topics. What is genetics? Genetics is nothing but the study of heredity and variations. We have defined heredity. We have defined variations and the two different types of variations. How the term, who coined the term, and what is its literal meaning. The origin of genetics, its significance, and different branches of genetics, namely Mendelian genetics, plant genetics, animal genetics, microbial genetics, molecular genetics, population genetics, radiation, quantitative genetics, cytogenetics, physiological, eugenics, euphenics, and finally genomics and proteomics. And today I'll go a little bit easier, slowly, so that you can write down the important scientific discoveries in genetics. The scientists who do research in genetics are called as geneticists. The scientists who do their research in genetics are called as geneticists. And let's see their contribution. The foundation stone of genetics was laid by, as we all know, it is by Gregor Johann Mendel, and he is actually an Austrian botanist who laid the foundation of science of genetics by his hybridization experiments. And he put forward these concepts, after which, after nearly more than 3 decades, 3 scientists discovered Mendel's principles of inheritance, and they were namely Correns, De Vries, and Schermark. About this we are going to discuss later. And these are the scientists who are called as rediscoverers of Mendel's laws. So these are the 4 scientists: Gregor Mendel and Correns, De Vries, and Schermark. De Vries belongs to Holland, Correns belongs to Germany, Mendel belongs to Austria, and Schermark belongs to what we call Austria itself, where Mendel is present at the time. And the concepts that were established after the scientific discovery of genetics is by William Bateson and his counterpart Punnett. William Bateson is considered as the British biologist who coined the term genetics. So Bateson is the one who coined the term genetics, and he translated Mendel's papers, which were in Austrian language, into English language. And the second important geneticist is Punnett. We know about monohybrid cross and dihybrid cross, where we come across with these squares. We call them as Punnett squares. And he is the one who discovered the fundamental concepts of linkage, sex determination, etc. along with Bateson, and he proposed the Punnett square model of representing genotypes in both monohybrid cross as well as dihybrid cross. So whatever the square that we use in mono and dihybrid crosses, that was said to be proposed by this Punnett only. So this is the square that he has proposed. So this is for monohybrid cross and this is for dihybrid cross, which we will discuss later. And next came the process where till that time only we have two kinds of scientists with respect to biology, botanists and zoologists. And after the discovery of this foundation stone being laid by Gregor John Mendel for genetics, these were known as geneticists. And coming to them, the foremost among them is Johansen. And he belongs to Denmark. He is a Danish botanist. He developed the concept of pure line. Please underline this one. Johansen is the one who developed the concept of pure line. What is a pure line? The true breeding individuals are called as pure lines. I will be explaining you this topic later. And Johansen is also the one who coined the term gene, genotype and also phenotype. So with respect to Johansen, you have 4 points. One is he developed the concept of pure line. 2, he coined the term gene. 3, he coined the term genotype. And he also coined the term phenotype. This is all about Johansen. Then came the most important scientist of modern genetics, and he is called as T.H. Morgan, Thomas Hunt Morgan. So Thomas Hunt Morgan is called famously as the father of experimental genetics. You can write it down. Thomas Hunt Morgan is popularly called as the father of experimental genetics. Mendel is father of genetics. T.H. Morgan is father of experimental genetics, and William Bateson is called as father of modern genetics. These three you have to keep in mind. One is father of genetics. Who is that? Gregor Johann Mendel. Second one is father of experimental genetics. That means a number of theories have been proved with respect to genetics by this scientist. And he is nothing but Thomas Hunt Morgan. And the third one is father of modern genetics. He is nothing but Bateson. Okay. And Thomas Hunt Morgan is an American zoologist who turned to become a geneticist. He is famous for his experiments on Drosophila melanogaster, and he proposed two theories. Number one, the theory of linkage. Number two, the theory of sex linkage. Sex linkage, you know, it is nothing but sex-linked inheritance, where there are three types: X-linked, Y-linked and XY-linked. Okay, and these two concepts, linkage theory and the concept of sex linkage, were proposed by Thomas Hunt Morgan. He is Thomas Hunt Morgan. And then came another scientist, C.B. Bridges. He is also an American scientist. He proposed the genic balance theory of sex determination. So there is a topic in your syllabus called as sex determination, and in sex determination, sex is determined by the balance between the genes present on autosomes and allosomes, which is called as genic balance theory. And that genic balance theory was proposed by C.B. Bridges, who also discovered gene duplication, the process of gene duplication in Drosophila. So Bridges can be attributed with two discoveries. One is genic balance theory. Number two, gene duplication in Drosophila. Okay, let's see who he is. And here is C.B. Bridges. And after these discoveries, so many experiments were conducted. It led to to the it led as a pathway to the development of new branches of genetics, which we have discussed yesterday. So biochemical genetics is evolved later, and radiation genetics also evolved by the experiments of different scientists. One pair of scientists is called as Beadle and Tatum. These two are Americans. They worked on inheritance of mutants in Neurospora, and it paved the way for the development of the branch of biochemical genetics, which is also called as molecular genetics. Neurospora crassa is a haploid individual. So because it is haploid, if it is exposed to some radiation, what happens? Some mutations occur and it becomes a mutant. And based on these mutants, certain concepts were established by Beadle and Tatum, which we'll discuss later. And with respect to Beadle and Tatum, we have to attribute the development of biochemical genetics or molecular genetics. And his experiments were done on Neurospora crassa. These are Beadle and Tatum. George Wells Beadle, Edward Lorite Tatum. And the next one is H.J. Muller. Muller, we can say that he is the one who demonstrated that X-rays speed up the natural process of mutation. So mutations, we know that Hugo de Vries proposed the theory of mutation, but the demonstration that X-rays can speed up the process of mutation was done by the experiments of H.J. Muller. Okay. Excuse me. Next, Mendel. We know Mendel proposed the concept of factors, and these factors were called as later by Johansen as genes. But no one knows the relationship between the Mendel's factors and the present day genes. And for the first time, there are certain scientists, mainly Sutton, who explained the relationship between Mendel's factors and chromosomes. So he is Mr. Sutton. He is American biochemist. He proposed the chromosomal theory of heredity. So what is Sutton famous for? He proposed chromosomal theory of heredity. Okay, and concluded that, or hypothesized that, chromosomes are the physical basis of heredity. Chromosomes are the physical basis of heredity. So what is physical basis? So if you say there is hydrogen, can you show it? No. Hydrogen combines with oxygen to form water. Can you show it? Yes, we can show it. So the formula H2O can be physically shown as water. Likewise, this is the scientist. He first proposed that for heredity, no one knows how we derive characters from parents. Now we can say that sperm and ovum fertilizes, it gives rise to a zygote, and zygote gives rise to an individual. But no one knows until the discovery of the microscope what actually inside the sperm and ovum is. Later we came to know that they contain nuclei. So nucleus is transmitted or transferred from parents to offsprings. So we can consider nucleus as the unit of heredity. But later, after the discovery of electron microscopes, etc., we come across with chromosomes inside the nucleus. And these chromosomes are passed on to the next generation during the process of fertilization. So we can now easily say that transmission of characters is done by the chromosomes present in our parents to the offsprings. Thus, these are the physical basis of heredity. Okay, so Mendel explained that there are factors which are responsible for heredity, and Sutton shows that chromosomes are the basis of heredity, and now we can combine these two. Factors present on chromosomes are the basis for heredity. Okay. Next came three more scientists, Avery, MacLeod and McCarthy. Avery, MacLeod and McCarthy. What did they do? These are the pioneers to demonstrate that DNA was the genetic material with their transformation experiments in bacteria. So these are the three scientists who demonstrated the famous reproductive mechanism of bacteria that is called as transformation. Okay. So by this transformation experiment, they proved that DNA is the genetic material. And this experiment was done in bacteria. It laid the foundation for the evolution of bacterial genetics. So evolution of bacterial genetics was said to be seen in the discovery of the transformation experiments in bacteria done by Avery, MacLeod and McCarthy, who demonstrated that DNA was the genetic material for the first time to the world. And coming to the evolution of molecular genetics. The pioneers among them are different scientists, Watson and Crick, Rosalind Franklin, Morris, Hugh, Frederick Wilkins. So these are the four different scientists who paved the way for the evolution of the molecular genetics. So what does they do? Watson and Crick were famous for the molecular structure of DNA. They proposed the double helical structure of DNA. We know that. Who is Rosalind Franklin? Rosalind Franklin is a English chemist. She's neither a botanist, neither a zoologist. She is just a chemist. Okay, a scientist of chemistry branch. She is doing her experiments on X-ray diffraction technique. X-ray diffraction technique, and it led to the development of DNA molecular structure and structure of virus by using X-ray diffraction technique. The same technique was adopted by Watson and Crick later to elucidate the double helical structure of DNA. And the third important one is Morris Hugh Frederick Wilkins. He is a British physicist. See, these are not biologists. Franklin is a chemist and Wilkins is a physicist. And he became later a molecular biologist. And he got the Nobel Prize, as do the other scientists. Their contribution is that so their research contributed to the scientific understanding of different phenomena. One is called as phosphorescence, isotope separation, optical microscopy, X-ray diffraction, and to the development of radar. And this X-ray diffraction studies of Wilkins were used first by Franklin, later by Watson and Crick, so that we could know the double helical structure of DNA. Okay. And coming to the modern gene concept, foremost among them is another female scientist like Rosalind Franklin. She is Barbara McClintock, and she proposed the concept of jumping genes, which we call them as transposons. So Barbara McClintock is famous for the concept of jumping genes or transposons. And later came another scientist, Seymour Beringer. Seymour Beringer is famous for modern concept of gene. Write down. He proposed the modern concept of gene, for which he used three terms. One is called as cistron, the other is called as recon, the third one is called as muton. So he coined three terms for gene. One is cistron, the other one is recon, the other one is muton. We'll discuss about this when we deal with the concept of gene. Next, Jacob and Monod, they proposed what we call it as operon concept in 1961. This operon concept explains a phenomenon called as gene expression and its regulation. So how a gene is regulated or how a gene's expression is regulated is said to be demonstrated by the experiment of Jacob and Monod in bacteria based on Lac-Operon concept in 1961. And then came Nirenberg and Matthaei. They are the ones who deciphered the genetic code. Nirenberg and Matthaei deciphered genetic code. And finally, our own Indian, Hargobind Khorana. He is an Indian born US scientist who prepared the first artificial gene. First artificial gene was prepared by H.G. Khorana, where in East, that too in the year 1970. So these are the different scientists who contributed for the development of genetics in earlier times. Now different set of scientists came forward, and with respect to the concepts that were evolved by those scientists, we'll discuss them in the coming classes. Coming to the terminology. Without knowing the basic terminology of genetics, we cannot understand what actually different concepts of genetics are. So let us discuss about these concepts or terms. First one is chromosome. Can anybody tell me what is a chromosome? Let it be a discussion. What actually you feel a chromosome is? Nobody is listening? Am I audible to everyone?
Yes, sir.
Good. What is chromosome?
It is a thread-like structure.
Thread-like structure. Present in? Where
Nucleus.
is present in nucleus? So according to Nagendra Kumar, chromosome is a thread-like structure present in the nucleus. Good. Any other?
A bundle of DNA can be coiled together, they form chromosome.
Good. According to 22 BS MBO 11 VSK, it's a bundle of DNA called as chromosome. Good. Any other? Any other definition?
Chromosomes are made up of proteins, sir.
Good. According to Bhargavi, chromosomes are made up of protein. Excellent. Any other decision and definition?
And it is a single molecule of DNA, sir.
Very good, excellent. It is a single molecule of DNA according to Yamini. Good.
Which is carrying two genes from the one generation to another generation is known as chromosomes.
Double excellent, which is responsible for heredity. Hence, we call chromosomes as vehicles of heredity. Good. Any other? Any other?
They carry coded genetic information that can be replicated or transmitted.
Very good, excellent. They carry coded genetic information that can be replicated for another generation. Excellent. Any other? Okay, we have discussed about chromosome. Can anybody tell me what is chromatid?
Condensed form of a chromosome.
Condensed form of a chromosome is called as chromatid. Good. Any other definition? Chromatid. Okay. Then what is DNA? Where is it present? In chromatid or chromosome? Or both?
Both, sir.
It's present in both. It's present in chromosome as well as it is present in chromatid. So instead of using the term chromosome or chromatid, why don't we simply call it as DNA? Why we are using these different terms? Can anybody tell me why we are using the term chromosome, chromatid, DNA when all are same? Huh?
Depending upon its stage
Depending
or its
upon
form
its stage or form, we call them as chromosomes, chromatids or DNA. Okay, let's see.
There exists in different types of organization, structural levels.
Very good. So coming to chromosome. What is chromosome? Chromosomes are rod-like or fiber-like structures made up of a nucleic acid and, as one said, a protein also. Not just somebody saying that chromosome is made up of DNA only. Somebody said that the chromosome is made up of protein only, but actually it's a combination of these two. So DNA is a nucleic acid. Okay. So here chromosome is a structure which is made up of nucleic acid, that is DNA, and also proteins that appears in rod-like or fiber-like fashion. So it's a rod-like or fiber-like nucleoprotein structure where actually it is present observed in a cell. Why we are not using the term nucleus? Because if you say nucleus, you restrict the definition only to eukaryotes which have nucleus. But whereas prokaryotes, there is no nucleus, but still too they have a chromosome. So you should not say that chromosomes are present in the nucleus. Rather we say that they are observed in a cell, so that both eukaryotes and prokaryotes can be brought into the same definition. Okay. So what is a chromosome? It's a rod or fiber-like nucleoprotein structure observed in a cell that is responsible for carrying the genetic information in the form of DNA. Whereas in prokaryotes, there is only one DNA molecule. That means only one chromosome. Whereas there are one to many inside the nucleus in eukaryotic cells. Okay. This is the basic definition of a chromosome. What actually chromatid is, chromomere is, chromonema is, and DNA is, we'll discuss in later class. Next, coming to the homologous chromosomes. So this is a plural name and singular name is said to be, we call it as homolog. And another one is heterologous or non-homologous chromosomes. So what is the difference between these two? So let's say a child is born with 46 chromosomes, and for this child there is a father and there is a mother, and mother got 46 chromosomes and father got 46 chromosomes. Father through his sperm contributed only 23 and mother through his ovum contributed only 23. That means the child received 23 from father and 23 from mother. Why is it so? When we see the first chromosome of the father, with respect to its length, with respect to its centromere position, with respect to its arrangement of genes, it is similar to the first chromosome of the mother. That means lengthwise it is same, position of the centromere wise it is same, and arrangement of the genes wise it is same. So thus both these are said to be same with respect to the genes, centromere and length, and such chromosomes are called as homologous chromosomes. Okay, and coming to this second chromosome, likewise the second chromosome is also lengthwise it is same, position of the centromere same, and arrangement of gene is also same. Let us say here is A, here is also, with respect to that, the gene responsible for the same character is also present. Okay, but when we compare chromosome 1 with chromosome 2, here it is metacentric chromosome, here it is submetacentric chromosome. That means based on the position of the centromere, they are different. And coming to the genes, here there are A, B, C, here there are D, E, F. That means based on the arrangement of the genes also they are different. And here this is very long compared to the second chromosome, which is short compared to the first one. Okay, so thus these are said to be not of the same height or length. In that case, they cannot be homologous, and we call them as heterologous or non-homologous chromosomes. Okay, let's see the definition. Homologous chromosomes are a pair of chromosomes. They are a pair of chromosomes whose corresponding loci are similar. So what is a corresponding locus? This is the first pair of chromosome, of which one is from father, which we call it as paternal chromosome. The other is from mother, which we call it as maternal chromosome. Thus we have pairs of chromosomes. One is paternal, the other is maternal. And I am saying corresponding loci are similar. What is corresponding loci? Here, when I speak of the first locus of the father, I say that this first locus or first position on the first chromosome of I received from father has gene for skin color. Then what happens? I receive the first chromosome from my mother, where on the position, first position, that is locus, there is gene for skin color. Second position, there is a gene for hair shape that I received from father, and mother also contains the gene for hair shape only. That means when we speak of first locus in this chromosome, we should speak of the first locus on its counterpart. Then these two loci are called as corresponding loci. The gene for skin color and hair shape are not corresponding loci. These two are corresponding loci, and these two are corresponding loci. When the corresponding loci are similar in a pair of chromosomes with respect to the character under consideration, those chromosomes are called as homologous chromosomes, of which one is always paternal, the other is always maternal in case of sexually reproducing organisms. Okay, is it clear? So what is homologous chromosome? Homologous chromosomes are a pair of genes. You can observe here also. This is the one locus, there is P2131. Here there is another gene P2131. And here this gene does not match the gene present here. Here it is Q12 and here it is Q12. That means these two chromosomes are said to be having the same gene arrangement. These two chromosomes are having the same gene arrangement, but these two are not having the same gene arrangement. In that case, here these are called as homologous chromosomes, and these are also called as homologous chromosomes, but these two are called as non-homologous chromosomes. Okay, and coming to the heterologous or non-homologous chromosomes. Sorry, another 2 points. Homologous chromosomes are found to be structurally similar, and they are observed because they are paired. We cannot see them in haploid individuals. They can be observed only in diploid and polyploid individuals. Okay. Example, first pair, second pair, third pair until 22nd pairs of chromosomes in human beings in females up to 23rd pair, which is XX also, that is homologous chromosomes. Coming to the heterologous chromosomes or non-homologous chromosomes. Any two chromosomes which are different, hetero means different, with respect to the gene's structure and centromere location, they are said to be called as non-homologous chromosomes. That means, example is chromosomes of two different pairs. The first one and second one are non-homologous. The second one is non-homologous to third one. And the third one is non-homologous to 21st one, and so on. In addition to this, in case of male, there are X and Y chromosomes which are also non-homologous. Okay. Next, coming to the locus, gene and allele. So what is locus? The position of a particular gene or a DNA sequence on a chromosome is called as locus. So this is chromosome I am speaking of. And each chromosome contains one DNA molecule. Okay, when I separate the chromosome from its into DNA and proteins, chromosome into DNA and proteins, this is DNA and the proteins are histones, which make up into what we call it as chromosome here. Okay, and this chromosome is having, according to what we call concept of linkage developed by Thomas Hunt Morgan, it has a linear sequence of genes which are arranged like the beads on a string. Okay, so there is a string we have taken and we have arranged beads, or you take a thread and tie flowers on it to make it a garland. So garland has so many flowers, and at the first position there is jasmine flower, then second position there is another flower. So the first position where the jasmine flower is present here we call it as locus. So here there is a first gene present here, and this is the first locus, and followed by another place where another gene is present, that is called as second locus. Thus a number of loci, locus is singular, loci is plural. A number of loci are present throughout the length of chromosome, where each locus is occupied by a particular gene. Okay, coming to with respect to DNA, how we'll define a locus? So we'll define a locus with respect to DNA. So what is DNA? DNA is a double helical structure. So when we separate the two strands, there are two strands, and both the strands have adenine, guanine, cytosine, thymine, etc., where the nucleotides are arranged in a sequential order. And the DNA sequence present here up to this point, whatever the nucleotides present there, they form into what we call a place where this forms into a particular gene, and we can call this as locus. Thus, with respect to chromosome, it is the position of a particular gene on a chromosome. With respect to DNA, it is a position on the DNA with respect to the specific DNA sequence. So thus, where the gene is present on a chromosome, we can call that position as locus. Okay. Now, second one, gene. So gene can be called as a unit of heredity. What is it responsible for? It is responsible for a particular character. So in famous Mendelian experiment, we know there is a gene for tallness, that is called as capital T, and there is a gene for dwarfness, that is called as small t, likewise, so on. And coming to the allele. So what are alleles? Alleles are the alternate forms of a particular gene present on the corresponding loci of a pair of homologous chromosomes. That means the term allele can be used only in diploid individuals, but not in haploid individuals, because haploid individuals have only one chromosome. That means here there is only one locus, and where the gene is present. And whereas in diploid individuals, there are pair of chromosomes, homologous chromosomes, and there are two loci and there are two genes which are called as alleles. So in other words, why we call them as alternate forms? Because let's say when evolution started, only tall plants are present. Only tall plants are present in Pisum sativum. So the first pair, let us say, there is a first pair of chromosomes. Here there is a gene for tallness and there is a gene for tallness. So how many loci are present? They are said to be two loci, and both loci are occupied by the same gene, that is gene for tallness. In due course of time, mutation has taken place, and on the same locus, what happened? One of the two genes modified to form small t, a recessive gene. And when this is present, the plant became a dwarfer one. And when the T gene, capital T gene is present, it is a taller one. So then what is the difference between these two? Here in the same species, we come across with two different genes that code for the same character, that is stem length. So thus, tallness and dwarfness are two different traits of the same character. Likewise, capital T and small t are two different genes that code for the same character, and these are alternate forms to each other. Hence, we call them as alleles. So, whenever you speak of alleles, alleles are always a pair of genes. Whenever you speak of gene, it is always single. Okay? And for you, I have a small question. I have a small question. If you have understood what gene is actually, what allele is actually, let me please answer this question. All genes are alleles. All alleles are genes. Which one of these is true? Is true or false? Which one is true and which one is false? If both are true, you can say both are true. If both are false, you can say both are false. If one is true, the other is false, you can say both.
All genes are alleles true. All alleles or genes are false.
Very good. Any other answer?
The first one is correct, sir. All genes are alleles.
All genes are alleles. That is true. All alleles are genes is correct. Any other answer? Nagendra?
Second one is correct, sir.
Second one is correct. Tanmay? Both are correct. Good. Second one is correct. First one is right. So,
Sir, second one is correct, sir. All alleles are genes, sir.
okay. So, those who say that the first one is correct, that means all genes are alleles, how will substantiate that statement? Bhargavi, you want to ask me something? You raised a hand? Okay. So why you say that all genes are alleles is correct? Who is going to support it, that statement? Okay, let me explain. I am saying two statements. All genes are alleles and all alleles are genes. I'll represent this here. Okay. So this is a first pair of chromosome. I am not going to answer. You are going to answer. There is a pair of genes for stem length. Okay. Capital T, small t. So capital T and small t, are they alleles or genes? Alleles or genes or both?
Alleles. They are genes.
They are only genes, not alleles. Good?
So both, sir.
They are both.
Yes, sir. They are alleles.
They are only alleles. Okay, leave about that. So next one, there is a gene for, say with respect to Mendel's concept only, or because you have already studied, I am asking you this one. Flower seed shape, round and wrinkled. So R and R, They are alleles or genes?
Both, sir.
They are both.
Sir,
Huh?
genes are
Okay, leave about this. Now,
character.
what about T and R? They are alleles or genes or both? And also
Genes, sir.
small t and small r.
Genes, sir.
They are genes. They are not alleles.
They are not of the same type. No, sir. They are not alleles.
Why?
Because one character will express the height. Because they are heterologous chromosomes.
No, these are homologous chromosomes. Heterologous means first pair of chromosome. This one is non-homologous to this one. These two are same. One is from father, paternal, the other is from mother, maternal. Hence, these are same and we call them as homologous chromosomes.
సార్ అలీల్స్ అయితే సేమ్ క్యారెక్టర్ని రిప్రజెంట్ చేయాలి సార్. జీన్స్ అయితే ఏ క్యారెక్టర్ అయినా చెప్తాయి కదా సార్. సో అక్కడ 2 క్యారెక్టర్స్ని చెప్తున్నాయి కాబట్టి అవి జీన్స్. అలీల్స్ అయితే ఓన్లీ 1 క్యారెక్టర్ కోసమే కదా సార్ చెప్పాలి.
Good. So as please go on.
Yes, sir. So the capital T is for it is indicating the length of the plant, whereas the capital R is explaining about the structure of the seed. So both are different, sir.
Okay. As Tanmani is good. So here, what is gene actually? Gene is a unit of heredity that determines a particular character. That means gene for tallness, gene for roundness. But capital T and capital R, they are genes, but they are not alleles because they are not coding for the same character. R is coding for seed shape, round seed shape. T is coding for stem length, tall stem length, isn't it? So then these never cannot be alleles. Why? Because one is present on one locus, the other is present on another locus. We can simply say that they are present on different loci. So when they are present on different loci, they can never be called as alleles. Okay? They are simply genes. Here capital T is a gene, small t is a gene, capital R is a gene, small r is a gene. But with respect to stem length character, the first chromosome that I have received from father, on the first locus, I have a gene for stem length, that is tall. The first chromosome that I have received from my mother, on the first locus, I have a gene for stem length, that is dwarf. Okay, leave about the dominant recessive relationship. I have received these two. Where they are present? They are present on the corresponding loci. That's what I am saying. What is a pair of homologous chromosomes? These are a pair of homologous chromosomes when the first locus is having the same gene for same character, and the first locus of this chromosomes also should have the same character for gene. Same gene for the same character. In that case, these two only are called as alleles because they code for the same character. They code for the alternate forms of a particular gene. That means one is for dwarfness, the other is for tallness, but they are said to be the alternate forms of the same character, that is stem length. In that case, capital T and small t are alleles, capital R and small r are alleles, whereas capital T and small r, because they are present on different loci, they are non-allelic in nature. So thus, capital T and small t, individually we can call them as genes. Together, we can call them as alleles. Likewise, capital R and small r, individually we call them as genes. Small r is a gene for wrinkle shape and capital R is gene for round shape. But together, with respect to the homologous chromosomes, we call them as a pair, and this pair is called as alleles. Thus, capital T and small t, or capital R, small r, are both alleles as well as genes. But with respect to when they are considered together, capital T and capital R, because they are present on different loci, because they code for different characteristics, they are not alleles. They are said to be just genes. Okay, so the statement, let us see once again. The statement goes like this: all genes are alleles. That means capital T, small t, capital R, small r are alleles. Is it true?
No, sir.
No. Why? Because capital T only are allelic pair. First allelic pair. And capital R, small r is another allelic pair. So these two are two different pairs of alleles. All are genes, but all these are not alleles. That means all genes are not alleles. Clear?
Yes, sir.
And the second statement is all alleles are genes. All alleles are genes. Let's see the first pair of alleles, capital T and small t. This is a pair of genes which are called as alleles. So can we call these alleles as genes? Capital T is a gene or not?
Yes, sir.
Yes. Small t is a gene or not? Yes. Likewise, in capital R, small r, these are called as alleles, and individually capital R is a gene, small r is a gene. That means all alleles are genes is a true statement, whereas all genes are alleles is a false statement. Are you able to understand?
Yes, sir.
So can you write it down in your notes? All alleles are genes. You write it like this: all alleles are genes, but all genes are not alleles. But all genes are not alleles. Okay, clear? Are you there?
Yes, sir.
Okay, good. So let's see this with this example. So these are a pair of homologous chromosomes. These are a pair of homologous chromosomes. And this is the first locus of paternal chromosome. This is the first locus of maternal chromosome. And the first locus are said to be having gene small a, and the second locus is also having gene small a. In that case, here these two are called as alleles. Okay? And the alleles can be same or they can be different. That means here it can be capital T and capital T in one plant, because I am a tall plant. I have received from my father tall gene, and also my mother is also a tall plant. I have received tall gene. So thus both the genes of an allele pair can be same. But here let us see, there is a dominant gene capital B here and recessive gene small b here. But both these code for the same characteristic. Thus they are alleles only. They are alleles only because they are present on the same locus. Let us say this is second locus and this is second locus. So because they are present on the same locus, they are said to be alleles. But alleles doesn't mean that they are always same. They can be different also. For example, I'm a tall plant. My mother is a tall plant. I have received tall gene. My father is a dwarf plant. I have received a dwarf gene. Thus, I can have different genes on the same locus which constitute an allele. Thus, allelic pair can have the genes which are same on the same or corresponding loci of a pair of homologous chromosomes, or the genes can be even different or dissimilar. Okay? And coming to the haploid and diploid condition, you know better. I need not explain this. A condition where an individual has a single set of chromosomes is called as haploid. For example, if I have only 23 chromosomes, 23 single chromosomes, then I am a haploid individual. Whereas if I have 23 pairs of chromosomes, that means if I have two sets of chromosomes, previously I have only one set, then I become a haploid individual. If I have two sets of chromosomes, then I'll become a diploid individual. If another one is added, I have a third set of chromosome, then I become a triploid individual. If another one is added, a fourth set of chromosomes, then I become a tetraploid. And above diploid, all are called as polyploid individuals. Thus, a haploid individual has only one set of chromosomes, diploid individual has two sets of chromosomes, and a polyploid individual has more than two sets of chromosomes. If there are three sets, we call them as triploid. If there are four sets, we call them a tetraploid, and so on, pentaploid, hexaploid, septaploid, octaploid, nonaploid, and decaploid, until 10 sets are present. Okay? Next, coming to the difference between the character and trait. Most of the students fail in distinguishing between these two. So that's the reason why I have put this here. So what is a character? So when we speak of a character, it is the feature that distinguishes an individual. Okay? So between these two individuals, there is a two plants. So the first one, what do you call this as?
It is a tall plant.
It's a tall plant, you call it as. And here this one we call it as a dwarf plant, isn't it? But by seeing side by side only, you are saying that it is tall and it is dwarf. But both when together, when we speak of this, we consider this as a stem length character. We consider this as a stem length character. So with respect to stem length character, it can be taller or dwarfer. So this distinguishing feature by which an individual can be identified is called as a character. Stem length, seed shape, eye color, hair shape, intelligent quotient, all these are said to be characters. Let us say Samuel sir is having an intelligent quotient of 20 and Mr. Nagendra is having an intelligent quotient of 40 and Tanmay is having an intelligent quotient of 120. So these are the varieties of this character. So these varieties of the characters are called as traits. Likewise, when we speak of stem length, there are two varieties. We can call them as variations or variants. So the variants of a character can be called as tall and dwarf. So these are the different features of character and trait, which this will I post in the group. You can study on your own so that you can understand it. I don't want to waste much time on this simple topic. Okay, next one is phenotype and genotype. What is the phenotype and genotype? When you see a person with respect to color, what you say? He is either black or white, either tall or short, you can speak. So the physical appearance of an individual is called as its phenotype. So why I am black? I am black because I have a pigment called as melanin in my body. So why this pigment is produced? Because I have a gene M which is responsible for the production of melanin in my body, which is recessive in other individuals where the amount of pigment is less, so that they are said to be very brighter in color than me. So in that case, here there is a physical appearance, black or white, which we call it as phenotype. And this physical appearance is because of the genes inside, which is called as the genotype. So let's see the difference between these two. Phenotype, it is the observable traits of an individual, which is determined by its genotype. I have a feature that is skin color. It is an observable trait. That means it can be seen. But the genes inside we cannot be seen. But that which can be seen is caused because that which is not seen called as genotype. And how we define genotype? Let's go for the passive voice again. So example, phenotype round seed is there, green seed is there, round yellow seed is there, green, likewise. So we are just telling what kind of this character is, then we can say it is phenotype. But how this round appeared? Because there are genes for roundness. How this green color appeared? Because there is a gene for green color. Why this is yellow in color? Because there is a gene for yellow color. So the genetic makeup of an individual which is responsible for its phenotype is called as genotype. So the genes responsible for our phenotype constitute the genotype. Okay. So that yellow color is due to the genes capital Y and capital Y, and the green color is due to the gene small y, small y. So yellow and green are said to be phenotype characters, and capital Y, small y give rise to, or the genetic makeup, that is genotype. Next, coming to the dominant gene and recessive gene. We can call them as dominant allele and recessive allele. Why? Because we are speaking with respect to a diploid individual. Okay, an allele that expresses its phenotypic effect in heterozygous state by suppressing the counterpart, that is recessive allele, is called as a dominant gene. For example, I have crossed a tall one with a dwarf one, and I have got a tall one. What is this tall? The difference between these two is this is a hybrid tall. This is a hybrid tall. We call hybrid individuals as heterozygotes, which we are going to discuss in the next slide. So in a hybrid, the character that is expressed is called as dominant character, and the gene responsible for the dominant character is called as dominant gene. And the character which is suppressed in the hybrid or heterozygote is called as the recessive character. The dwarf character is recessive, and the gene responsible for recessive character is called as recessive gene or recessive allele. So dominant gene is said to be represented by capital Y and capital Y for yellow color. Okay, so it is the dominant gene. Capital Y is dominant over small y. And coming to recessive gene, it is an allele whose phenotype is not expressed in a heterozygote. That means in the cross between tall and dwarf, I have got only tall plant, and in this tall plant the dwarf character is suppressed. That means this gene, small t gene, is said to be suppressed by the capital T gene, which is dominant, and hence the small t gene which is not expressed in the hybrid or heterozygote is called as recessive allele. Okay, next, coming to the homozygosity, heterozygosity and hemizygous nature. What is homozygous? So let's take again a pair of alleles, a pair of chromosomes. So these are a pair of homologous chromosomes, and I am taking the first locus here. So first locus in Tanmay. So with respect to blood groups, Tanmay's blood group is, let's say, B group. So she has genes IB and IB. Okay, and another person, Purnima, she has genes IA on the same first chromosome but in another individual. And likewise, I have taken another third person, Samuel sir, first chromosome only myself, and the first locus I have IA, IB. So what is the difference between these three? Here the pair of alleles are same in the first individual and in the second individual. In such conditions, they are called as homozygous individuals. Homo means similar, zygous means pair. Okay, so here when we consider a pair of genes or alleles, when both genes present on the corresponding loci of a pair of homologous chromosomes are similar, then we call this condition as homozygous condition. But with respect to myself, I have received one gene IA from my father, another gene IB from my mother. Thus, both the genes of this allelic pair are not same. The genes of allelic pair are not same. In that case, we call it as heterozygous individual. Okay. So what is homozygous? A condition where a pair of genes or alleles present on the corresponding loci of a pair of homologous chromosomes is similar. Then this condition is called as homozygous. And both the genes can be dominant genes, for example capital T. Both the genes can be recessive, small t. Thus homozygous is homozygous dominant or homozygous recessive. Okay, let's see the second one. Heterozygous, a condition where the genes, two genes or alleles present on the corresponding loci of a pair of homologous chromosomes is dissimilar. Hetero means different. Zygous means pair. So when we consider this pair of genes on the same locus, one codes for tallness, the other codes for dwarfness. In that case, we call this as heterozygous individual. So with respect to seed color, the heterozygous is said to be capital Y, small y. In a heterozygous individual, always there is a dominant allele and a recessive allele, and the dominant suppresses the recessive. Hence, heterozygous are always dominant. Homozygous can be dominant or recessive, whereas heterozygous is always dominant. And finally, coming to the third one, hemizygous. When we consider the 22 pairs, the first two 22nd pair, all are said to be called as autosomes in human beings. In female, the 23rd pair are X and X. So these are also same. Thus females have all homologous chromosomes only. But when coming to the male, the first 22 pairs are said to be homologous, whereas the 23rd pair, we have X chromosome which is long and a Y chromosome which is short. And the genes present here are not present here, and the genes present here are not present on the X chromosome. In that case, when you see this locus on X chromosome, there is only one gene. So you have studied about color blindness. So color blindness in male. So for color blindness, there is a gene called as capital C, which is for normal vision, small c for color blindness. And here this male carries only one gene, either for normal vision or for color blindness. Thus there is only one gene, the other gene is not present on the Y chromosome. And that condition, this condition is called as hemizygous. Hemi means half. Zygous means pair. So out of this pair of alleles, only one is present. Hence we call this condition as hemizygous condition. So we can define it as like this. A condition in a diploid individual where only one copy of a gene is present. Here two copies are present, here two copies are present. In homozygous, both the copies are same, either dominant or recessive. In heterozygous, both the copies are different, and it is always dominant only, because recessive cannot suppress the dominant gene. In hemizygous, we can either have the dominant gene, we can either have recessive gene, but never both, because the corresponding locus is absent on its counterpart. And such genes are called as hemizygous genes. So that is the difference between homozygous, heterozygous and hemizygous condition. So coming to this example, homozygous, capital Y is homozygous, small y is also homozygous, capital Y is small y is said to be heterozygous. And with respect to hemizygous nature, there are X-linked genes on X chromosome in male and Y-linked gene present on Y chromosome in male. Okay, and before going to this, I have a question for you. I am representing here three conditions. Okay, please explain whether it is homozygous or heterozygous. With respect to seed shape, which is round and wrinkle. So there is first plant with capital R and capital R. Is it homozygous or heterozygous? Smaller, smaller.
Homozygous.
So how you distinguish between these two now? Both are homozygous. How you differentiate between these two?
It is dominant, it is recessive.
So first one is called as
Dominant.
homozygous dominant, and the second one is called as homozygous recessive. Very good, excellent. And coming to the another representation where there is a capital R gene, small r gene.
Heterozygous.
Heterozygous dominant or recessive?
Heterozygous.
Only dominant, sir.
Heterozygous is dominant. So I have a statement for you. I have a statement for you, true and false. You have to tell me which one is correct. Homozygous individual is always dominant. Statement number 1. Statement number 2. Heterozygous individual can be dominant or recessive. Which one of this is true?
Both are false. Both are false.
Both are false. Why? Because homozygous individual can be
Individual is dominant or recessive. Heterozygous individual is always dominant, sir.
is always tense. Hence both are said to be false. Okay, very good, excellent. So with this we conclude the basic terminology of genetics, and few more terms are there. For example, one term, that is operon, I missed. Can anybody tell me what operon is?
A particular gene code for a protein.
A particular gene codes for a particular gene coding for lactose is called as operon. Okay, lac operon. Okay. So
Sir, it regulates the transcription.
very good. So write down what is operon. So when you see operon, there are 3 genes, Z, Y and A. Before that there is a regulatory gene. Before that there is a promoter gene. Okay, and all these genes are responsible for utilization of lactose or glucose in the medium. So when I have glucose in the medium, I take it and use it for my energy. And likewise bacteria take up lactose or glucose in the medium and use it for energy. But actually this lactose is present outside the bacterium. So first of all it has to be taken up inside, for which it has to pass through the plasma membrane. So there must be a protein which can carry the lactose inside, and that is said to be produced by one gene. Before going to that, this lactose has to be added with an acetyl group, unless otherwise it cannot be carried. So this acetylation is done by another gene. After going inside, lactose is broken down into glucose and galactose molecules. That is done by a galactosidase enzyme, and that is a protein, and that is produced by this gene. Thus there are 3 genes responsible for the utilization of lactose. Okay, and these are regulated. Whenever lactose is present, they are said to be transcribed. If lactose is absent, they are said to be not transcribed. In that case, they are regulated by the different genes present adjacent. So all these form into a cluster. we can call it as a complex. We can call it as a complex. So what is the difference between a what is the difference between a normal gene and operon? Normal gene is single. It is only one gene responsible for one or many characters. If it is a pleiotropic gene, is responsible for many characters. If it is a normal gene, it is responsible for only one character. Whereas operon is a group of genes or a cluster of genes or a complex of genes responsible for the expression of, responsible for a particular character or particular metabolic activity. Lactose utilization is a metabolic activity. In that case, this cluster of genes is called as a operon. Write down operon. Operon is a group of genes present in prokaryotes responsible for a metabolic activity. Okay. So with this we conclude two topics now. One is, today we have discussed about the different scientists that contributed for genetics and the basic terminology of genetics. And in the next class, we'll discuss about the Mendel's laws. Okay. So before that, I request all of you to come up with, by studying the entire topic of Mendel's laws, because you already have listened this topic one or two times, one in the intermediate, the other time during your degree, because you are finally students, you already have heard it. So in that case, I need not have to explain the entire process of Mendel's laws. So I will be explaining first basically the experimental design of the Mendel. So why Mendel has taken Pisum sativum, what are the different techniques that were employed by Mendel, like emasculation technique, okay, self-pollination technique, followed by reciprocal cross technique, back cross, test cross. All these techniques we'll discuss tomorrow. And after that, I'll be discussing two things. One is, actually, when can somebody tell me who proposed Mendel's laws? So we say that Mendel's laws, as the name says, they are proposed by Mendel's laws. Newton's laws were proposed by Newton. Okay, likewise Mendel's laws were proposed by Mendel. But actually Mendel hasn't proposed any law at all. Mendel made only 4 deductions. What does Mendel do? Mendel made only 4 conclusions based on his experiments. And those 4 conclusions were made into laws by the rediscoverers of Mendel, especially Correns. Okay, so thus Mendel did not propose any law. He only proposed the principles of inheritance in the form of 4 conclusions. So we are going to discuss about 4 conclusions made by Mendel and the 2 laws proposed as Mendel's laws by rediscoverers of Mendel, especially Correns, and the critical review of Mendel's laws. What is critical review? So Mendel says there is a dominant recessive relationship. Is dominant recessive relationship is always there. Mendel says there is segregation. Is segregation true for every living being? Mendel says there is independent assortment. Is independent assortment possible for every characteristic present on the in all the living organisms present on earth or not? So these are the few questions that I am going to ask you tomorrow in the first class itself. After that, we'll discuss about the complete Mendel's laws. So the first thing is Mendel's principle of dominant and recessive universal? Number 2, is segregation possible with all the characteristics or genes under consideration with respect to the different living organisms? And is independent assortment a universal phenomenon? So these are the 3 questions which you have to answer before tomorrow's class, so that be thorough with the entire Mendel's laws topic, and we'll have a discussion, not a class tomorrow, we'll have a discussion. Okay.